Mendel bred many pea plants and he studied them and found patterns. And some of the patterns he found were that traits were passed from parents to offspring. Some variations of those traits were dominant over other variations of traits. He also found that traits were passed down independently of each other. So a tall pea plant would not necessarily pass down its color traits with its tallness. It would, the color trait and or what he called factors, the factor for seed color and the factor for plant height were inherited independently of each other. We're going to look now at how we can apply some of Mendel's principles. Our objectives for today are first to explain how probability can be used to predict inherited traits, to explain how alleles segregate when more than one gene is involved, and lastly, to identify Mendel's contributions to our understanding of genetics. Probability is the application of mathematical principles to the study of how likely events are to occur. If you flip a coin, what is the probability of the coin landing on tails? Well, if you flip one coin, the probability of getting tails is 50%, or 1/2. If you toss a coin 3 times, what is the probability of getting tails 3 times in a row? Well, the first is, in order to do that kind of math, we multiply the probability of getting tails the first time times the probability of getting tails the second time times the probability of getting tails the third time. So we do 1/2 times 1/2 times 1/2, and we would find that the probability of getting tails 3 times in a row would be 1/8. Being independent means that the outcome of one coin toss does not affect the outcome of another coin toss. So no matter how many times you flip the coin, the probability of the next toss landing on either outcome is 50%, or 1/2. You could get 100 tails in a row. The next time you flip the coin, you have a 50% chance of getting heads. Here we see a combination of individuals in the F1 generation. This could be through self-pollination or it could be through cross-pollination. Remember, the P generation was the true breeding population. The plants in the P generation had only dominant or only recessive alleles. And when we crossed a dominant and recessive plant from the P generation, we got the F1 generation. So both, well, here we see two F1 plants. All F1 plants have the same set of alleles. And they exhibit the same traits. And they all exhibit the dominant trait. We don't see any recessive traits in the phenotypes of the F1 generation because they all have one dominant and one recessive allele. So we see a bunch of green plants. We breed them together. And then we get this ratio on the bottom, 3 green plants for every 1 yellow plant. The homozygous dominant and heterozygous allele combinations produced 3 green plants, while the homozygous recessive allele combination produced 1 yellow plant. These quantities follow the laws of probability. If you crossed a homozygous dominant plant with a heterozygous plant, would the offspring be green or yellow? Well, even if you had a heterozygous plant in the parentage, you would still get all green plants because you would never, from that cross, you would never get 2 recessive alleles. Each time a gamete is produced, remember plants produce lots and lots of gametes. each time a gamete is produced, it has a 50% chance of getting the first allele. In this case, we see it's a dominant allele, and it has a 50-50 chance, or it has a 50% chance of getting the recessive allele. So each event, each time a gamete is produced, it's a random event. It's always a 50-50 chance. So the probability of a gamete receiving a dominant gene or a dominant allele is 50% or 1/2. The probability of a gamete receiving a recessive allele is 50% or 1/2. And the probability of green offspring is 3/4, because those heterozygous individuals also come out green. And the probability of yellow offspring is 1/4. There are two possible genotypes of a green plant. When you see a plant that's green, you don't know from looking at it what its genotype is. You only know its phenotype is green. So what are the two possible genotypes? You can have homozygous dominant, or you can have a heterozygous genotype. The phenotype of a plant that has two alleles for yellow color is always going to be yellow. So if you see a plant exhibiting the recessive characteristic, you know its phenotype. But if you see it exhibiting the dominant characteristic, then you don't know what its genotype is unless you go and study its lineage. So how can we fill in these blanks? Two organisms may share the same phenotype, but have different genotypes. It doesn't work the other way around. If you know the genotype, you know the phenotype. But if you know the phenotype, you don't necessarily know the genotype. I've been using these terms, heterozygous and homozygous. Homozygous means that the organism has two identical alleles for a gene. You can have homozygous dominant or homozygous recessive. Heterozygous means that you have two different alleles for a gene. So if we look at these sample plants, which of them are homozygous for the height characteristic? I'm sorry, this is not showing height. Which of them are homozygous for the color, the pod color characteristic? And which of the plants is heterozygous for the pod color characteristic? We see that plants 1 and 4 are both homozygous. 1 is homozygous dominant, 4 is homozygous recessive. And 2 and 3 are both heterozygous. One of the best ways to predict the outcome of a particular cross is to use a model called a Punnett square. Punnett squares use mathematical probability to help predict the genotype and phenotype combinations in genetic crosses. We start our Punnett square by identifying the alleles each parent could pass on to the offspring. Here we see an example of two ospreys. Those are the type of bird. They're both heterozygous, so they both have one dominant allele and one recessive allele. So we can see that the gametes that are possible to be produced for each bird will produce 50% dominant alleles and 50% recessive alleles. We indicate that on this organizer called a Punnett square, this model called the Punnett square. And so since we have 50% dominant and 50% recessive alleles, we have two different columns. So we put one dominant allele and one recessive allele on each, or one dominant allele on the first column and one recessive allele on the second column. We do the same on the rows because we have 50% dominant and 50% recessive alleles. We then fill in the columns and the rows to match the header of the column or the row. So we have a capital B here, so we're going to put a capital B in each box below that capital B. And we have a lowercase b here, so we're going to put a lowercase b in each box below that lowercase b. Likewise, we fill in the rows. So we have a capital B here, so we're going to put a capital B in each box in the first row. And we have a lowercase b for the second row, so we're going to put a lowercase b in each box on the second row. And this shows us the ratio of possible genotypes of the offspring of this pairing of two heterozygous individuals. So how many different genotypes do we see? We see 3 different genotypes. We see all the variations of genotypes, but not in equal proportions. We also see 2 possible phenotypes, because remember, a heterozygous individual is indistinguishable from a homozygous dominant individual. What is the probability that these offspring will have the dominant phenotype? Well, the probability for having the dominant phenotype is 3/4, because all 3 of these out of 4 have the dominant phenotype. And then the probability that an offspring from this cross will have the recessive phenotype is only 1/4, because there's only 1 out of 4 that shows the recessive, the homozygous recessive genotype that is needed to exhibit the recessive phenotype. It's possible to look at more than one characteristic at a time with a Punnett square. When you focus on 2 characteristics at a time, it's called a dihybrid cross. In a similar manner as in the previous slide, see if you can walk yourself through this diagram for making a Punnett square using a dihybrid cross. In the gamete formation, we have to see all the different combinations. We have 2 different traits. So we have size, a trait for size, and a trait for pod color. So when we look at all the different combinations of gametes that can be produced from two hybrid individuals, we see that you can have capital T, capital G, capital T, small g, small t, capital G, or small t, small g. Therefore, when we make our Punnett square, we have to include 4 rows and 4 columns, because we have 4 possible gametes from each parent. From that point on, the process is exactly the same. You fill in the column to match the headers of the column, and you fill in you fill in the rows so that they match the headers of the rows. And then you end up with 4 alleles in each organism. Because you have 2 traits, you have 4 alleles. And then from here, you can see there are more variations of genotypes that you can have exhibited. And of course, there are also more variations of phenotypes, because you have 2 traits that you're studying. After Mendel showed that alleles segregate during the formation of gametes, he still wondered if the segregation of one pair of alleles affected another. To find out, he followed 2 different genes for P color and P shape from one generation to the next. This Punnett square represents a dihybrid cross because it involves 2 different traits, seed shape and seed color. Round is dominant over wrinkled, and yellow is dominant over green. How is the genotype of the offspring different from that of the homozygous parent? The offspring are heterozygous for both traits. What is the phenotype of each parent? And what is the phenotype of the offspring? Well, one parent produces round yellow seeds. We have to name both traits. And the other parent produces green wrinkled seeds. So the offspring in this case all produce round yellow seeds because the offspring are heterozygous. But what if you had to predict the genotypes and phenotypes in the F2 generation? Draw a Punnett square for a cross between the F1 individual. So draw a Punnett square for two hybrid plants, two heterozygous plants for both traits, and figure out the genotypes and phenotypes that you would see in the F2 generation. Will it be possible for there to be homozygous recessive offspring in the F2 generation? And how do you know? Well, you'll find the results when you do your Punnett square. Where are the homozygous recessive offspring? I guess this answers the last question. There are homozygous recessive offspring. Can you find this in the diagram? They're right here. One out of 16 is a homozygous recessive for both traits. How many different phenotypes do you see present in the offspring? Well, in this case, you see not 2, but 4 different phenotypes. You see round and yellow, you see round and green. So here, well, you can see, you have round and yellow, most of them are round and yellow. You have what? 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. 9 out of 16 are round and yellow. Then you have 3 out of 16 that are round and green. You have 3 out of 16 that are that are wrinkled in yellow, and then only 1 out of every 16 in the F2 generation is wrinkled in green. What's the probability of a cross of these parents, these hybrid parents, the F1 generation? What's the probability of a cross of these parents producing a fully homozygous recessive offspring for these two traits? It's 1/16. And what proportion of offspring are likely to have the phenotype dominant for yellow and the recessive for wrinkled seeds? Count the number and put the numerator over 16. What proportion of offspring are likely to have the phenotype dominant for round and recessive for green seeds? And how many different kinds of gametes could this individual produce, and what are they? All of these answers are shown on the Punnett square. What proportion of offspring are likely to have the dominant phenotype for both traits? What ratio of phenotypes does this Punnett square predict? This is a ratio. To fill in the ratio, you're going to put the numbers of yellow round seeds to the number of green round seeds to the number of yellow wrinkled seeds to the number of green wrinkled seeds. So what evidence did Mendel have that alleles segregated independently in the cross shown here? Well, the evidence he was able to cite is that all combinations of the phenotypes resulted from this dihybrid cross of individuals from the F1 generation. What would have happened? What phenotypes would have resulted if the alleles did not segregate independently? In other words, if the dominant R and dominant Y genes always stayed together, and the recessive R and recessive Y alleles always stayed together. Well, in that case, you would get round yellow seeds and wrinkled green seeds. So he found that each of these traits sorted out independently. Remember, Mendel didn't breed, he didn't just get 16 plants. He got many, 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 many plants. So this is showing a ratio, not an actual number. So independent assortment, this is a law, a principle. This is a principle that Mendel defined. Independent assortment says that genes for different traits can segregate independently during gamete formation. In this case, this was demonstrated by the 9:3 to 3:1 ratio of phenotypes. Inheritance is determined by units called genes, which are passed from parents to offspring. Where more than one form of a gene for a single trait exists, some alleles may be dominant and others recessive. Remember that Mendel's principles of heredity were observed through patterns of inheritance, and they form the basis of modern genetics. Mendel's principles apply specifically to sexually reproducing organisms. So organisms that produce gametes, that includes flowering plants and animals and some other organisms. So I try to predict the terms that complete these sentences. Each adult has 2 copies of each gene, one from each parent. These genes segregate from each other when gametes are formed. Alleles for different genes usually segregate independently of each other. That's called independent assortment.